Good afternoon. So before I start, can I have a show of hands in the room for the people who has worked on the iron colloid drug products or thinking about doing that? Well, that's a lot of people that add a lot of pressure to me, I guess. Okay, so first, thank you for the organizers for inviting me to this workshop. It's my pleasure to present a discussion on the physicochemical characterization and impurity consideration of the iron colloid drug products. So this is just a standard disclaimer. The presentation reflects only the views of myself and should not be used against the FDA's views and policies. Obviously, FDA's views and policies controls. So for today's presentation, we will start with a short discussion or introduction of iron colloid drug products. We will move to FDA's recommendations for this type of drug products. We will discuss the physicochemical characterization and impurity considerations, and we will then go over a few case studies and finally wrap up with the conclusions. So here's a table listing a class of iron colloid drug products on the US market. They are presented by the trade names, the non-proprietary names, the current applicants, the approved dates, and also physicochemical properties here represented by particle size. These drug products all contain iron and carbohydrate. Depends on the drug product. We may have different iron oxides, and carbohydrate may be natural products or synthetic derivatives. One important feature is the particle size of the drug products. They are in the nanometer range. That is the reason why they're called colloid system, because they are stable suspension with very small particle size. So let's first discuss the common characteristic of this class of iron colloid drug products. They are all iron replacement products for the treatment of iron deficiency anemia. Here I must emphasize that what we discussed today are focused on this class of drug products, and their main routes of administration are the IV injection. There are other iron carbohydrate drug products on the market for other indications with different route of administrations. And it is important to recognize that they are not within today's topic of today's discussion. For this class of products, the drug product is in general composed of iron oxide core and carbohydrate shell. By formulation design or by the nature of the interactions between the carbohydrates and iron oxide core, there is more or less the presence of free carbohydrate components. In terms of mode actions of delivery, the commonly accepted theory involves a step of internalization of iron colloid complexes, which is followed by the incorporation of elemental iron to the internal iron pole. This group of drug products is known to have some adverse effects. For example, hypersensitivity reactions such as anaphylaxis or oxidative stress caused by low molecular iron species. So today's workshop is for complex generic drug product applications and development. Therefore, it is appropriate for us to ask the following question. Can this class of iron colloid drug products be approved via ANDA approval pathway? And the answer is yes. In this slide, I provide you a partial but direct copy of FDA draft guidance on Ferumoxytol, one of the iron colloid drug products. FDA has published draft guidance on this class of drug products. In fact, one for each drug product. And the agency's current thinking for bioequivalence involves three components: in vivo BE studies, in vitro particle size distribution analysis, and special considerations. And here the special considerations is copied, pasted on this slide. So two important items for special considerations. First, the requirements of Q1, Q2 sameness. And the second, the established establishment of sameness in physicochemical properties. So let's dive deeper on the physicochemical property and characterization. There are three areas of considerations. First, the physicochemical property characterization should be considered on a product-by-product basis. For under applications, the efforts should aim to establish pharmaceutical equivalence between the ROD and the generic version. And the properties under consideration should be those likely affecting the safety and efficacy of the drug products. However, as we will see in the next slide, iron colloid drug products are complex drug products which contains inorganic and organic components. These components interact with each other to form this, in my opinion, dynamic mixture. Very often multiple physicochemical properties need to be analyzed and compared, and different analytical techniques are often required for the analysis. Therefore, a well thought out and systematic approach is highly recommended. Here I recommend a review article published last year by AAPS. This article summarized literature reported techniques for the physicochemical characterization. More importantly, it offered a strategic approach to the characterization and provide discussions on the analytical method, validations, and data interpretation. It is, in my opinion, a great article to reference if you're interested in characterizing this iron colloid drug products. A key strategy, according to this article, is to divide the physicochemical properties into three categories. Those associated with the whole particle, those associated with iron core, and those with carbohydrate shell. Among each category, relevant properties are considered and analyzed using appropriate analytical techniques. In this slide, some of those characteristics and commonly used tests are listed. This approach provides clarity and a system to tackle many properties associated with the drug product. Please note that the properties and tests I list in this slide are by no means the exhaustive or the minimum list of the properties one should consider. This is just a demonstration of the properties one strategic approach rather that one can take. For each specific drug product, we recommend a risk-based design of experiments. For example, if the iron core is composed of different iron oxide species, then the content of each iron oxide would be of high interest. On the other hand, if the carbohydrate shell is made of modified carbohydrate units, then perhaps the modification pattern on the carbohydrates or the ID of the substitution groups would need to be analyzed as well. Likewise, impurity considerations can also be divided into three categories. Here I would like to offer two thoughts which are often overlooked. First, low molecular weight iron and free iron species are not known to involve in internalization. Basically, they are not involved in mode of actions. Therefore, they should be considered as impurities and treated as such. The second, potential degradation products produced by carbohydrates, either via the manufacturing process or during stability or during storage, I should say, should be monitored and controlled as well. In addition, as we often discussed, that because the manufacturing process-related substance and impurities do not add value to the quality of the drug product, they should be controlled at levels comparable to those observed in the reference product or according to ICH recommendations. All right, let's take a look at other important considerations. First, I would like to reiterate that based on FDA draft guidance, sameness in certain physical chemical properties between RD and the test product should be established. To achieve the sameness, a comprehensive approach is preferred. In other words, from a quality standpoint, we would like to gain understanding of the product based on the whole picture, the totality of the drug package, the data package. We try not to focus on one datum or one set of data to draw conclusion. Such a totality approach, in my opinion, will also help to battle the variability issue one may encounter. By formulation design or by nature of the drug product, free carbohydrate is present. In the same time, it is possible that low molecular weight iron species present in the drug product as well. Those components may interfere with the characterization. Therefore, it is important to consider removing those interference species prior to the testing. According to the labeling information, certain in-use and dilution conditions are applied to the drug product. As such, in-use and dilution studies should be considered for physicochemical characterization. Selection of appropriate analytical techniques and method procedures are highly important, and we will discuss that further in the next slide or two. The important characteristic for the iron colloid drug product physicochemical properties is the visibility or variability in characterization data. The variability may come from multiple places. Multiple places. The inherent variability of the drug product, for example, can be a contributing factor. In addition, variabilities can be caused by analytical techniques, by sample preparation, experimental conditions, or by using different analytical techniques for the same parameter. All this add to the challenge one faces in the characterization efforts. So how do we proactively minimize or try to minimize the variability in the data? We strongly encourage the characterization of multiple batches. The information would allow us to assess the reproducibility of the manufacturing process. It should also help enhance our confidence in the accuracy and precision of the data package. We recommend the adoption of the same sample preparation procedures, conditions, and data analysis so that the inter-procedural variabilities is reduced to a minimum. As you will see in the case studies, if the data obtained are variable, an effective strategy to mitigate the risk is to use orthogonal or complementary techniques. Sufficient sampling size and proper statistic analysis also will help reducing variability. Finally, I would like to emphasize the importance of proper method validation and use of suitable reference standards. We often see the firm submitting comprehensive data package, however, with no method validation information. The information cannot be properly assessed because without this validation information, we have no idea how the method performed. Given the variability we are dealing with, it is important that the firm provide method validation information or calibration information for the analytical methods used for physicochemical characterization efforts. All right, with those discussion points, let's go into the case study. So the first case study, the firm proposed to conduct comparative total iron content testing between the test product and the RLD. And the assessment is the total iron content may include colloidal iron and the low molecular weight iron and free iron species. From the discussion, you will know that we believe that the low molecular weight iron and free iron species are considered impurities. Therefore, when the firm combines the colloidal iron and the low molecular weight iron, it may allow actually the interference of impurity for the content. Therefore, we recommend the firm to measure the colloidal iron contents and control low molecular weight iron and free iron as impurities. Comparative studies should be designed to report the above as separate properties. Let's go to case 2. The firm proposed to dilute the iron sucrose test product 1,000 times prior to conducting particle size measurements via DLS. Well, we understand why the firm provided this proposal because the drug product has an intense color due to the iron 3 species. And the color may interfere with the particle size measurement under concentrated condition. However, when the dilution is conducted, one should also consider whether it would cause the iron complex or the iron carbohydrate complex to unravel. we know that the carbohydrate and the iron core, they actually interact with each other, not really via covalent bonding or ionic bonding, but weaker bonding. Therefore, when you dilute, it is possible that the carbohydrate will dissociate, and that may cause the reduction of the particle size. In the same time, when the carbohydrates dissociate, the iron core may actually aggregate, and that may actually in turn increase the particle size. Therefore, the assessment is that the proposed dilution may impact particle size measurement. And what we propose to the firm is to conduct sequential dilution studies. Basically, you start from the concentrated conditions and you dilute 10 times, 20 times, and 50 times, and go on. So that way you can see during the dilution process whether you see the particle size change one way or the other. All right. Case study 3. So before we go to that, first let's take a look at the left panel. This is a graph that copied from the article quoted below. So this is basically a XRD diffractograms of the iron colloid drug products. As you can see, some of the drug products actually produce rather sharp peaks, for example, Ferumoxytol at the bottom. But other drug products, for example, iron sucrose, the second one, they produce rather weak and broad peaks. The firm proposed to use XRD to determine the crystallinity and particle size distribution of iron sucrose iron core. Well, although XRD is known and is probably commonly used for this type of task, for this specific drug product, the weak and broad diffraction peaks, as I shown on the left side, may not be suitable for crystallinity ID and particle size distribution determination. Therefore, we recommend the firm to use orthogonal techniques to corroborate the finding they have in the XRD experiment. So, hopefully with this discussion, I give you an impression that we have discussed the physicochemical characterization and impurity consideration for iron colloid drug products. We believe that orthogonal approaches, including physicochemical comparability of the drug product, considered in combination with Q1, Q2 sameness, coupled with PK equivalence, ensure the bioequivalence of the iron colloid drug products, which would allow the generic version of the drug products to be approved via ANDA pathway. So with that, let's go to the conclusion. So today we discussed that ANDAs for iron colloid drug products are reviewed on a product by product basis. Sameness in physicochemical characterization is one of the key components for establishing equivalence. A comprehensive approach, risk-based design of experiments, and proper validation calibration are required to reduce data variability. Finally, I would like to acknowledge my colleague, Dr. Andre Rao, Dr. Binkai, Dr. Pahala Simarmora, and Dr. Panzu for their contribution and discussions for this presentation. And thank you for your attention.